All right, everyone. Today we're going to be talking about genetics, so let's go ahead and get started. So first of all, heredity is the transmission of characteristics from parents to offspring, and genetics is the study of how DNA passes through generations. A term we're going to use a lot is characteristic or trait. And these are things that are typically either proteins or influenced by proteins, as we'll see in a minute. All these things have to do with DNA. Proteins dictate almost all of our physical traits. Well, how does that work? The order of the DNA letters, called bases, tells the cell how to make the protein. It's basically a cookbook. In a cookbook, you have many recipes. Each recipe is made of letters arranged in a certain way, and depending on how these letters are arranged, they spell out different instructions. The instructions tell you about a product that you'll be making. In the same way, DNA bases are like letters. Depending on their arrangement, they will tell the cell how to make a specific product, in this case, a protein. Oh, I need to change my pointer options pen. There we go. Yeah, so here's the DNA. Oh, it's working, and there's the protein. Excellent. So why are DNA and proteins important? Well, a gene is a segment of DNA that contains instructions for making a specific protein, as you may remember. To continue our analogy, a gene would be like a single recipe within a cookbook. Here we have 3 genes attached to one another, and each gene tells the cell how to make a different protein. Then each protein goes and does its job within the cell. For example, the protein melanin will produce skin pigment. An allele is a variation of a gene. So, for example, for the gene eye color, we have 2 variations, or 2 alleles, blue and brown. To continue our cookbook analogy, for the gene cookies, we would have different variations or alleles, such as sugar, chocolate chip, or snickerdoodles. Pause the video and answer the question, and then resume the video. The answer is D. This poster shows the different chromosomes in the human genome. We have chromosomes 1 through 22, then X and Y. Each chromosome has different genes on it. This poster lists just a few of the genes on each chromosome. Let's take a look. I want to make very clear that there are thousands of genes on every chromosome. Only a few are shown here because there is not enough room to list them all. One of the genes is for cleft palate. You can see this baby has cleft palate. It's not a life-threatening condition and can be fixed with surgery. The gene for cleft palate, which is a segment of DNA, has a particular order of DNA letters. These letters spell out instructions for a product, which is the protein that causes cleft palate. In this way, the order of DNA letters dictates physical appearance. Whoa. Okay, there we go. Sorry, I've got a script, I'm reading it, and it was out of order. Anyway, another example is the sex-determining region Y gene, which makes the SRY protein. Here is a picture of the SRY protein. This gene is found on the Y chromosome. Each male of the human species has the genes necessary to develop ovaries and a uterus. In fact, we are all female by default. If the SRY protein is present, it will bind to the genes that cause ovaries and a uterus and inactivate them. This will then trigger the development of a male reproductive system. So what exactly do I mean when I say DNA letters or bases? Well, think about the English alphabet. We have 26 letters, and depending on how many are used and how we arrange them, we make words with different meanings. In the same way, DNA has 4 letters for chemicals in its alphabet. Depending on how many of these bases are used and what order they are arranged in, they spell out instructions for different proteins. It's very important to note that this is how a cell stores information, is in the order of these bases. A stands for adenine, that's the chemical adenine. T stands for thymine, there's a thymine. G stands for guanine, there's guanine, and C stands for cytosine, right there. You can look at the sequence of any gene. For example, here's part of the gene for the SRY protein, and this will tell you the order of these chemicals. Now for some more vocabulary. When we're talking about genes and how they relate to physical appearance, we use the word genotype to stand for the gene responsible for a trait, that is, the particular order of DNA letters. We use the word phenotype to describe the physical appearance resulting from this genotype. This is the trait for hairline shape. Notice there are 2 alleles. Each gene has a slightly different arrangement of DNA letters, but for the most part, they are the same. This is the genotype. It's the DNA. The phenotype is the physical result of the genotype. It's what you see. Here's another example. The phenotype is what you see. This is the phenotype of cleft palate. The genotype is the order of DNA letters, that is the actual gene. And for the SRY protein, here's the genotype, and here's the phenotype. Go ahead and answer this clicker question. And the answer is A. Today, we're going to discuss several kinds of genetics. First, let's get into Mendelian genetics. This kind of genetics is named after Gregor Mendel, who was the quote-unquote father of genetics. He was a European monk who bred garden peas and observed their offspring, and he came up with some conclusions. And in order to talk about his conclusions, we're going to need some more vocabulary. Cross means to mate two organisms. So, for example, I am the result of a cross between my mother and my father. Progeny means offspring. The P generation is for the parents. F1 stands for the first generation of offspring, so the offspring of the P generation. And F2 stands for the second generation of offspring, so the offspring of the F1 is the F2. Mendel's work drew several conclusions. First, that variations in characteristics within a population are caused by variations in genes. Second, that for each gene, an organism inherits two alleles, one from each parent. In this diagram, you can see that both the mother and the father contribute one chromosome to form a homologous pair. In this case, the gene is represented by the letter A. The allele from the egg is uppercase A, and the allele from the sperm is lowercase a. You can also see in this image Mendel's third conclusion, which is that a sperm or egg carries only one allele for each trait. Mendel's next conclusion was that alleles are either visible or not visible as a phenotype. That is, a dominant allele is expressed, meaning you see the result. A recessive allele is not expressed, meaning you do not see the phenotype. Let's imagine that the gene A is for hairline shape. We know there are two phenotypes for hairline shape, widow's peak and straight hairline. If we designate the widow's peak allele as a dominant allele and the straight hairline as a recessive allele, then we know that if these two alleles appear in an individual, you will only see the dominant one, meaning the widow's peak. This individual also has an allele for straight hairline, but you would never know by looking at this person because all you see is the dominant. Here's a clicker question. Pause the video and answer. The answer to this is D. Mendel also came up with what's called the law of segregation. This states that pairs of alleles separate during meiosis. So here's a pair of alleles, and here's a pair of alleles. And they're separating during meiosis, as you can see. And then they separate again. And that each gamete, these are the gametes, receives one allele or the other. So this is a pretty straightforward conclusion. He also led to the development of the law of independent assortment. And you may remember this from our discussion of meiosis. This builds on the law of segregation. Remember, the law of segregation states that pairs of alleles separate during meiosis. And the law of independent assortment states that they separate independently of one another. Which allele ends up in which gamete does not influence which allele goes into which other gamete. These are a couple of animations that you can watch, and I think they will be very helpful if you choose to watch them. Mendel's conclusions led to the development of the chromosome theory of heredity, which states that genes are located on chromosomes, and each gene occupies a specific place on a chromosome. In this image, the genes P, A, and B are on these chromosomes. This is the locus of gene P, this is the locus of gene A, and this is the locus of gene B. Recall that homologous chromosomes are ones that carry the same genes. Chromosome 1 from your mom and chromosome 1 from your dad together form a homologous pair. You get 23 chromosomes from your mom and 23 chromosomes from your dad. And resulting, you have 23 pairs, that is, 23 homologous pairs of chromosomes. Here we have a homologous pair of chromosomes. We know they are a homologous pair because even though they are different chromosomes, they carry the same genes. Alleles are represented by letters. A dominant allele is always represented by a capital letter, and a recessive allele is always represented by a lowercase letter. You always use the same letter for both alleles. For example, if green pea pods are dominant to yellow, then we use a capital G to represent the green allele and a lowercase g to represent the yellow allele. What we cannot do is assign a different letter to each allele because they are both the same gene. We cannot assign capital G and lowercase y. No, they either have to be both G or both Y. Here's some practice to give each member of the following sets of traits a letter. Pause the video and assign a letter to each. And now I will go ahead and do it. So for example A, smooth peas and wrinkled peas, what am I going to use? I will use the letter Q. And this is the dominant allele, so I'm going to put a capital Q. And then over this side, we have the recessive allele, so I'll put a lowercase q. Yellow peas, what letter? How about S? S, capital S for yellow, and then lowercase s for spotted. And whenever I'm writing alleles that are lowercase, I'm going to try and remember to put, oh, I forgot on that one too, to put a little line over them, because especially if the letters look similar in capital and lowercase, it's really important to be able to designate which one is lowercase. All right, and letter C, sweet peas and bitter peas. I'll use the letter B, capital B for sweet peas because it's dominant, and lowercase b for bitter peas because that's recessive. Combinations of alleles are called different things. Remember that you have two alleles from each trait for each trait, one allele from your mom and one allele from your dad. If both of these alleles happen to be dominant, we say you are homozygous dominant for that trait. If both of these alleles happen to be recessive, we say you are homozygous recessive for that trait. Here's an example. If purple is dominant to white, what is the genotype for a homozygous dominant flower? So a homozygous dominant flower would have two dominant alleles, so capital P, capital P. Homozygous recessive will have two recessive alleles, so lowercase p, lowercase p. There's a clicker question. Pause the video and answer. The correct answer is C. Here is another clicker question. Pause the video and answer. And the correct answer is A. Heterozygous means that the two alleles for a trait are different. If you get different alleles from each parent, we say you are heterozygous for that trait. So if purple is dominant to white, the genotype for a heterozygous flower is going to be having one dominant and one recessive allele. The phenotype is the physical result. And so we know that it's going to be purple because there's a dominant allele here. So we're definitely automatically having the dominant phenotype, which is purple. You're gonna have to forgive me. I'm trying to write with my finger on this screen. It's not going well. All right, clicker questions. Here's the first one. And here's the second one. The answer for number 1 is B, and the answer for number 2 is A, sweet. It is not sweet and bitter, even though the genotype has a dominant and recessive allele, because we're talking about complete dominance. So as long as there's even one dominant allele, it just takes over and you don't see the other one at all. So our phenotype is going to be sweet. Let's summarize and put it all together with this pair of homologous chromosomes. The genotype for this locus is capital P, capital P. That's homozygous dominant. For this locus is lowercase a, and that's homozygous recessive. And then we have our heterozygous, capital B, lowercase b. Here's some review. In flower color, purple is dominant to white. What are the phenotypes for the following flowers? Also label them homozygous dominant, homozygous recessive, and or heterozygous. Okay, so purple is dominant to white. So if purple is dominant, that means we're going to represent purple with a capital letter and white being recessive with a lowercase letter. Here in this genotype, we have two dominant alleles. So that's homozygous dominant. We have here a dominant and recessive. So that's heterozygous. And we have here two recessive alleles. So that's, oh, I can't even do this, HOMO, homozygous recessive. As for phenotypes, letter A has both dominant, so it's going to have the dominant phenotype, which is purple. So A is going to be purple. Letter B is heterozygous. It doesn't matter that there's a lowercase, that there's a recessive allele here. All that matters is the dominant allele. So we see the dominant phenotype, and that is purple. So B is going to be purple. And C only has recessive alleles, so that's going to show the recessive phenotype, which is white. Number 2, what are all possible genotypes for the following flowers? Well, A, purple, all genotypes just have to have at least one purple allele. The other one could also be dominant, or it could be recessive. Each of these genotypes will give you a purple flower. And for white, because it's recessive, we can't have any dominant alleles present, only recessive alleles. So that will give you a white phenotype. All right, Punnett squares. A Punnett square is a diagram used to show all possible combinations of gametes. So if you know the parent gametes, then you can know all the possible genotypes of the offspring using a Punnett square. What you do is you draw a box like this, and you put parent 1 genotype on one side, and you put parent 2 genotype on the other side. And then within this box, once you solve the problem, you will find all the possible genotypes of the offspring. And because you have all the possible genotypes of the offspring, you also know their phenotypes. Let's try a monohybrid cross. Monohybrid crosses look at one gene. For example, let's see, I have some hair above my knuckles. It's hard to see, but it's there. So that's the dominant allele, and having smooth knuckles is recessive. There are 3 possibilities for our genotypes. These are genotypes. We have homozygous dominant, homozygous recessive, and heterozygous. So let's go ahead and do this example. We're going to use the gene A, and we're doing a cross of a homozygous dominant and a homozygous recessive. So I'm going to choose the homozygous dominant parent to be on the top, and I write 1 allele above each column. And then I'm choosing the homozygous recessive allele to be on the side, and I put 1 allele in front of each row. Oh, I've got to put my lines. So now what we're going to do is we're going to take this capital A and draw it on all of the boxes that are in that column. And we're going to do the same thing with the other capital A. We take it all the way down, draw it on all the boxes that it's on top of. And then we're going over here to this side A, this lowercase a, and we're going to draw it on all of the boxes in that row. So we're just taking it all the way across. And the same with this A. We're just taking it all the way across. We're drawing it in all of the boxes in that row. So genotypic ratio, when you're asked to calculate a genotypic ratio, what you're calculating is the amount of homozygous dominant genotypes to the amount of heterozygous genotypes to the amount of homozygous recessive genotypes among the offspring. Okay, so these are the offspring genotypes. How many homozygous dominant would there be? Well, homozygous dominant is capital A, and there are no capital A in the offspring. So, whoa, we say 0. And then we put a colon. Heterozygous, that's capital A, lowercase a. There's my line. And there's 4 of them. 1, 2, 3, 4. So my second number is 4, and then I put a colon. Homozygous recessive is lowercase a, and there are none of those in this box. So then I put the letter 0. So 0 to 4 to 0 is my genotypic ratio for this cross. For the phenotypic ratio, when you're asked to calculate that, you're asked to calculate how many show the dominant phenotype to how many show the recessive phenotype. So in this case, the dominant phenotype is going to be whatever is designated by the capital A. I don't know, let's, whatever it is, it doesn't matter because we know that all of these are going to have the dominant phenotype because they all have a dominant allele. So that's 1, 2, 3, 4. So we write 4. And then recessive, there's none that are going to display the recessive phenotype. So that's 0. So 4 to 0 is our phenotypic ratio for this cross. Now let's do a heterozygous crossed with a heterozygous. Let's use P's this time. Let's say that big P is purple and lowercase p is white. So for heterozygotes, we're going to have one dominant and one recessive allele in each combination of alleles. And I'm going to put one parent on top and one parent on the side. So we've got our genotypes there. And then we do the same thing we did before. I'm going to start with this allele, this capital P, and write it in all of the squares that it is on top of. Then I'm going to do the same with the lowercase p and write that in all of the squares that it is on top of. And then I'm going to do a similar thing with this capital P. I'm going to write it next to all of the boxes that it is next to. So notice that the capital goes before the lowercase, just like if you're writing a sentence. Then I'm going to take this lowercase p across, lowercase p, there we go. And now these are all the possible genotypes of the offspring. And because we know their genotypes, we also know their phenotypes. Let's find their genotypic ratio. How many have the homozygous dominant ratio? Homozygous dominant is capital B, capital P, and that is 1. This one here is homozygous dominant. So our first number is 1. How many are heterozygous? That's capital P, lowercase p. Well, we have 2 that are heterozygous. So our second number is 2, then a colon. And homozygous recessive is lowercase p, and we have 1 of those. So our genotypic ratio for this cross is 1 to 2 to 1. Phenotypic ratio, how many display the dominant phenotype? Well, that's going to be 3. This one is going to display the dominant phenotype, this one, and this one. And we know that because they have dominant alleles. So first letter is 3. And then lastly, how many will display the recessive phenotype? Well, that's just going to be the remaining 1. So 3 to 1 is our phenotypic ratio for this cross. Go ahead and do a practice, and I will show you the answer in a minute. Also, you should probably keep in mind that this might be on the quiz at the end. All right. So if we assume that brown eyes are dominant to blue, a man and a woman are both heterozygous. They're both capital B, lowercase b. That's my line. So we're going to do a cross between 2 heterozygotes. And let's just do the cross right here. A capital B, lowercase b, capital B, lowercase b. So for the first one, we have capital B, capital B, capital B, lowercase b, capital B, lowercase b, and lowercase b, lowercase b. The expected genotypic ratio among the offspring. Remember, the genotypic ratio is going to be homozygous dominant to heterozygous to homozygous recessive. So homozygous dominant, we have 1. Heterozygous, we have 2. And homozygous recessive, we have 1. So our genotypic ratio is 1 to 2 to 1. I do not know what happened there. The expected phenotypic ratio is how many have the dominant phenotype to how many have the recessive phenotype. So the dominant phenotype is brown eyes. How many of these kids are going to have brown eyes? Well, the number that will have brown eyes is the same as the number that have the dominant allele. And so we've got 3 of those. 1, 2, 3. So 3 is our first number, and then we have 1 left. So that must be the second number. So 3 to 1 is the phenotypic ratio for this cross. Keep that in mind when you've taken the quiz. Maybe write it down. Dihybrid crosses. These guys examine the inheritance of 2 genes at once. And this demonstrates the law of independent assortment, which you may remember from the beginning of the lecture, states that alleles separate independently of one another during meiosis. So up to this trait, we've been discussing, up to this point, we've been using monohybrid crosses, which examine the inheritance of 1 trait. What if we want to track the inheritance of 2 traits at once? That's called a dihybrid cross. And using dihybrid crosses, we can demonstrate that alleles of different traits separate independently of one another. In this diagram, you see that there are blue alleles and there's green alleles. And at the end of meiosis, we have 4 gametes right here. The law of independent assortment states that any allele, any of these alleles, has an equal chance of ending up in any of the 4 gametes at the end of meiosis, and that this doesn't depend on where another allele might end up. None of them have to end up in gamete 1, 2, 3, or 4, or whatever. The alleles sort themselves independently of one another. The final location of one doesn't depend on the final location of another. Here's a clicker question. Pause the video and answer the question. And the answer is C. So for this example, let's do a dihybrid cross. Let's use hairy knuckles and the ability to roll your tongue. Take a second to check out whether you've got hair above your knuckles. I do, and I can also roll my tongue. So here's how we're going to demonstrate this. Beware the common pitfall. I'm getting ahead of myself. Let's cross 2 individuals that are heterozygous for both traits. So capital H, lowercase h, capital R, lowercase r. And cross that with capital H, lowercase h, capital R, lowercase r. Right there. Oh my God, how do I pause this? Oh shit. Help. No. Yes, I want to keep. Pause recording, please. Not stop presenting. Oh God, this is so embarrassing. Okay. Okay, well, stop, I guess.